DNA fingerprinting. DNA stands for deoxyribonucleic acid. It is called as the polynucleotide chain. A polynucleotide chain is double-stranded in human DNA. It contains two strands of DNA. One of the important applications of the Human Genome Project is the DNA fingerprinting. According to this concept of DNA fingerprinting, the technique of using DNA fragments resulting from restriction endonuclease enzyme cleavage to identify particular individuals is called DNA fingerprinting. The technique of using DNA fragments. These DNA fragments, which are obtained from the human genomic technique by the aid of the restriction endonuclease enzymes. Restriction endonuclease enzymes, which are the molecular scissors. Restriction endonuclease enzymes. These are considered as the molecular scissors and are importantly present in human genome sequencing. Whenever you use this technique of using DNA fragments to identify particular individuals, this is called a DNA fingerprinting. This technique is a worldwide technique used throughout the world, and due to its wide applications. DNA fingering technique was developed by Eric Jeffrey. Eric Jeffrey was a scientist who first of all developed this DNA fingerprinting technique in 1985 at Leicester University, United Kingdom. Inheritance of DNA is very stable. Every person has specific pattern of DNA sequence which shows a combination of DNA sequence of both mother and father. DNA, what comprises the genes in the human beings or other animals, they are unique in a particular person. That's why the scientists have put forward a theory, Thomson and Morgan. According to this theory, the two individuals are not exactly alike. No two individuals in the world are not exactly alike. If the genes and the DNA of even the two identical twins is 99% similar, but still there is 1% difference that makes 1% different from the another, or one individual different from the another. That's why we say every person has specific pattern of DNA sequence which shows a combination of DNA sequence of both mother and father. Study of DNA fingerprints help us in establishment of identity of a person, identification of criminals in case of a murder or rape, and paternity test in case of dispute parentage. For example, in court cases, we are having parental disputes upon one child. How can we identify the particular mother of that very child if there is a parental dispute? Such crimes exist in our society. Whenever these cases are referred to the court cases, how does the court define? The court defines the paternity and maternity of that very child depending upon this DNA fingerprinting. So these are very important techniques used in disputed parentage cases or criminal cases. It helps in identification even when the stains on victims' clothes are even several years old. How these are retained? In case of a murder, when the stains of the blood, stains of other things remain on the criminal's clothes or criminal's body, they can be detected after a long period of time. This is how we identify the criminals in our society. In India, first DNA testing fingerprinting was done in June 1989. This is the year when in India first DNA fingerprinting was done for setting a dispute parentage in Madras. There was a dispute parentage of a child in a court in Madras that was detected. His or her parentage was detected by this DNA fingerprinting in 1989. Laboratory for DNA fingerprinting is situated in Hyderabad at Center for Cell and Molecular Biology. CCMB. The headquarters of this DNA fingerprinting is located CCMB, Center for Cell and Molecular Biology, what we in short term call as forensic laboratory. Now, the forensic laboratory in India has several branches associated with the police departments of various states in India to detect these criminals. Paternity dispute cases are much more common in India, and most of them are referred to CCMB for DNA evidence. That is why this molecular biology has a separate branch that is called as forensic science. This forensic science is very important these days to detect the criminals or in case of a parental dispute. This is how we detect, this is how it is very important, DNA fingerprinting, how it is very important in the solution of criminal cases. But there are certain techniques involved in this. The first technique is VNTR, Southern blotting technique of fingerprinting, what we call as VNTR, variable number of tandem repeats. Variable number of tandem repeats. This is one of the important processes involved in DNA fingerprinting. That's all for this introduction of DNA fingerprinting.